Hello everyone. Let's start now. In today's class, we will discuss a few things that we have already discussed in the last class, and then a few more. So, some parts of this lecture are going to be the next assignment that is going to be graded. This lecture will be useful for that. So I would want you to pay attention and try to see if you can, if you have any questions, you're always welcome to ask. Is my audio clear? Somebody can confirm so that I'll start.
Yes, sir, it's clear.
Okay, good. So in the last class, we had a question about discussing gene synthesis. Well, the chemistry of it is not so important for us at this time, but it will be helpful if we have some idea about it. Just the general process, as I was already telling you in the last class. Here you have the 3' OH, and this is the 3' OH, and here is the 5' phosphate, which would have been, and it is blocked. These are the two reacting groups, 3' OH and 5' phosphate, which should have been there. And the blocking of these two reactive groups is important, and then you can synthesize how you want to do. So the 3' OH, as you're seeing in the second figure, you can see that you want to encourage the interaction or the formation of a bond between 3' OH and the 5'. It should have been phosphate, but they have, in chemistry, they have it differently. I'm not going into the details, but it has a certain group here, which also has the phosphate here attached to it. Chemically, they would have synthesized like this or added. They have activated. They have an activating group to it, and then so. Now this 3' OH is ready for activation, and the 5' of the second, the first nucleotide is blocked here. This is blocked, and the 3' OH would have had a glass bead, which is also blocked. So they would have unblocked the 5' agent here, or the carbon group here. And now this 3' OH of the second nucleotide is activated. And then using some catalyst, they would increase they would make this the phosphate bond form. And these will be processed further to make a DNA. And therefore, and slowly, in the next case, they would have removed this glass bead. They would have activated it and then keep on adding. That is how they would have done. 1 second. I think there is a mistake here. It is happening in reverse direction, isn't it? So this is the glass bead. You can assume that that is the stationary phase. Okay, this is the first nucleotide, and now the second nucleotide is added, and then they would have unblocked this 5', and they would have added another base here. That would have been the third base. That is how chemically DNA is synthesized. This is very different from how it would have happened in biological system. We don't need to know all this stuff, but it's good to know these things. So I was discussing about the adapter. I started off with linker, and then we went off to the adapter. There is a difference in linker and adapter. Here is a sequence, and say, for example, there are 3 more bases. I will discuss that. Why do we have those later? And represent any of the nucleotide. So this is the restriction site for PAMH1, I guess, and therefore it would have you can attach something like this to a piece of DNA molecule, on you can do the same on either side. Okay, something like that. And once you have added, you have to treat this with the restriction endonuclease so that it will get cut, and now you have overhangs that are of required sequence or and so on. That is about linkers. That means you have to treat it with a restriction endonuclease. So sometimes you may have a BAMH1 site within the region or the gene of interest. And if that is the case, you will not be able to treat it with BAMH1, or if you do, the gene of interest may also be cut, and we don't want that kind of things. Therefore, if we are obligated, that means the vector is having a BAMH1 site, and the fragment is also having a fragment as a gene of interest. I'll just erase this. Vector is also having a BAMH1, sorry, fragment is also having a BAMH1 site, but you want to clone using these ends. And how can I have BAMH1 overhangs here? We can always, I hope you remember, we can polish these and make it into blunt ends and then do blunt end ligation using T4 DNA ligase. Sometimes maybe that's not possible, and then we should be able to ligate these two at BAMH1 site. In that case, what if we can add some fragments that already have the overhang just like that, that represent BAMH1, and those nucleotide sequences are called as adapters. It is simple that we have the pin and the plug adapter. So to do this, I just want to say, for example, we have GGA, T, CC, and here is TA, GG. And if I cut it, it should have been cut in somewhere here, and this part should have been gone. So if I want to do that, the adapter overhang should have been something like this. Right. So this is the DNA should have been, the adapter would have sequence like this. So this would be 5 prime, 3 prime, and 5 prime, and 3 prime. If I take the same and reorient it, I would have this on the top, say for example, G, A, T, C, and C, and then there is G, and then you have another DNA, or a little more, something like this. 5 prime, 3 prime, and 5 and 3 prime. I didn't do anything other than I kind of inverted this into this direction, right? When you're taking many adapter molecules like this, and if I also have added a DNA of interest here, okay, this is the gene of interest that I want to add the adapters. So I have 3 components. Assume that I have multiples of adapters, and I have some fragments. And if I add ligase to this, the most probable ones that would actually get ligated would have been something like this, that because G, this can be, you can assume this fragment or this adapter can ligate here, G, A, T, C, right? And that would have another C and G is here. And if we get like this, then the whole concept of using the adapters itself is useless. We could have used linkers instead, because if we add ligase, it would have sealed these, and then we should have had to add another again a restriction diation with the restriction enzyme, which we actually wanted to do, which we want to prevent, because the fragment has the same restriction sites within itself. Therefore, we need to prevent that, and to prevent those, these people have used 5' OH, right? That means the 5' here that is there would have been, I'll try to see if I can get it off. Yes, the 5' here, it was actually a phosphate group. This was a phosphate group. Here you have a OH, and here is another OH. And because they have made it into OH, so if I remove that and this, and it would have been, here is a OH and here is another OH. Now, even if these two bind through hydrogen bonds, ligase will not be able to make a phosphodiester bond because there is 3' OH and 5' also has a OH, not phosphate. So you actually dephosphorylated the 5' OH. It can be done by many ways using alkaline phosphatase, or maybe when designing it chemically also, people can get rid of it. And so now you have a blunt end here, and there is a blunt end here, and here is another blunt end, and here is another blunt end. So what are all the possibilities? If we look at it, we will find that we will have the fragment, and then we will have, let's think of all the possibilities. So one side bound, but the other side, the adapter has not been bound. You can also have the fragment with the adapter on the left side, but not on the right side. You may also have fragment ligation like this, and also circularized products and so on. You may also have adapter products, something like this, just the adapters binding on the other side, on this side, okay, as I'm trying to show here, not on the overhang side, because on the overhang side, the phosphodiester bond cannot be formed because we have dephosphorylated the 5' phosphate. So this is another possibility where an adapter can dimerize. But anytime you have the sticky ends or the overhangs, they have much more higher probability than the blunt end ligation. Obviously, you can see that they are cohesive ends. They can attract and form hydrogen bonds, which is much more stable for the ligase to act upon. And the actual product that we are looking for is a product which has the adapters on both the sides. Okay, I'll just put it like this. Sorry. Something like that. This is the product that we are having. If we did not dephosphorylate the 5' phosphate, more of these would have formed. And if these more of the adapters like self-ligation have formed, then lesser this product, the actual product that we are looking for, would have happened. And that's going to be a trouble for us. I hope you understood the logic of why we are dephosphorylating here, and when we are trying to ligate it with the vector, what can we do is we can use polynucleotide kinase, and ATP is the substrate for it. It will transfer the gamma phosphate from ATP to the 5' of the adapter here, and then it can be used for ligation. Okay, so the enzyme is polynucleotide kinase. I hope you also know about ATP. Say I'm not drawing A, and then you have the ribose, and this is the 5' CH2, and then you have alpha, beta, and then gamma phosphates. The gamma phosphate is transferred to the 5' group, and that is how you can rephosphorylate the 5' ends so that they are ready for ligation into the vector. This is the vector. Okay. So we will learn some things like TA cloning. today. It is called as TA cloning for a simple reason that the fragment has a extra A site, and the vector, if we can generate a vector with T, just an open one T overhang of one base, which is just T. First thing is that when we are trying to do PCR, which we will learn today in the light of primer designing. So every time we perform PCR. Okay, sorry. So there is a strand, and this is the second strand. And if we are trying to do amplify this using Taq DNA polymerase, what it does is when it is amplifying, the product actually is not blunt-ended. Instead, on the 3' side, it has an additional 1 A added to it. I am not sure about the biological significance. I don't think there is one, but anyway. So this is a 3'. This is a overhang of one base pair, 3' overhang of one base, and that is adenine. And that is done by Taq polymerase in PCR. So what if we can directly manipulate this and clone it into a vector? That's the idea of it. So there is an enzyme called as HPH1. We might have to find if there are any other restriction endonucleases that might be useful, that might produce a T. The idea is if I had a vector like this with an overhang of 1 T, that is complementary to the overhang that we have on the amplified product. Then we can just directly put in there and add ligase so that it will get cloned. We don't need any restriction or processing of the PCR amplified fragment is not there. It is directly usable. So the simplicity of it is attractive, and in the sense processing the gene of interest. But this is a little bit tricky to get the overhang of T. There is one enzyme that is called HPH1. It is a type 2S. It recognizes this 5 bases, something GGT, GA, and then it cleaves something. You can count 1, 2, 3, 4, 5, 6, 7, 8. After 8 nucleotides, it makes a cut here. And on the bottom strand also it makes a cut. It is somewhere 7 bases around here. Okay, what it usually, we have to find places where we have softwares that can do this for us. You have to know the restriction site, where it is located. That can be predicted by the software. And then we can predict if there is a T on the 8th position. If there is one, then we know that it is going to generate a T overhang, such as shown here, right? So if we have a vector, say, for example, this is a vector like this, you would have cut it, found such a way that a T overhang is generated. And this can be now, the PCR product can be purified. Purified in the sense you have unused primers. There might be primer dimers and other things. So PCR purification kit is usually used. You can also do electrophoresis and cut out the fragment that is of actual length, and then directly add the PCR product to this, and then add a ligase, and it would have sealed it up. Now we have a recombinant plasmid. Okay. Yes. Now, today's actual focus is that this one particular one. What to do, say I have a gene of interest here, and I want to clone it in an expression vector. We will discuss about vectors later, but otherwise, expression vector, and there are cloning vectors. Cloning is to just make multiple copies of it, whereas expression is to get the ORF produce a protein. So it should produce a protein. And if it is an expression vector, then you need a promoter. Promoter is usually designated in this broken arrowhead like this, right? And you should you should not have it in the ORF should not be like this. Every time you point this way, this is the start and this is the stop codon. The ORF reads from here to here. That means when the mRNA is produced, it should have been produced like this. It would be 5' here and 3'. Start codon is here and stop is here. That is how you would have produced a protein, because the ribosome always reads from 5' to 3'. Therefore, you will get the correct protein. But if you had in this direction, say, I'll write, you have the promoter like this, and the ORF is something like this. Sorry. The ORF is like this. The transcript produced is the antisense of the mRNA. I think I will just take a minute here. So you have two strands. I would usually do it in the beginning classes, but I think I forgot, but I will recall. You have a start codon, okay? And this is, I'm writing in terms of DNA, so, and then here it is TGA. Now, what is this top strand called as? What all ways can it be? Is it a plus strand or minus strand? Is it a coding strand or a non-coding strand? Is it a template strand or a non-template strand? We should have these recorded well, or we should remember those things properly. Assume this is the mRNA, and you would have AUG and UGA. Now, what we should, this is mRNA is the plus strand, because it is having the codons in the readable fashion. It is the coding strand, again for the same reason. And it is also called as the sense strand. Now, which of these strands in the DNA, okay, this is the DNA, which of the two strands is looking similar to that of mRNA? The top one or the bottom one? The top one is similar, except for the U replaced with T. Everything else is same. This would have been the plus strand. It would have been the coding strand. It should be the sense strand. Okay, this we are talking in times in light of how information is processed during translation. What about when it is transcription? Which one is the coding strand, the top one or the bottom one? If top one was the template, okay, during transcription, the mRNA that would have formed would be complementary to it, right? That means it is actually, it will not form AUG just for discussion's sake, okay? But it would have been complementary to it. Something like that. So the top strand would have been the non-template strand in terms of transcription. So get this terminology right. I try to remember in this fashion. I try to write mRNA as the plus strand. It is the coding strand. It is the sense strand. And anything complementary to it would have been the minus strand. It would have been the non-coding strand. And it would have been the antisense strand. And in terms of transcription, because the promoter is here, the promoter, the RNA transcriptase, or what you call RNA polymerase, would actually read this strand and produce the complementary of this template so that it will get mRNA with this kind of orientation, AUG and UGA. Okay, so keep those things in mind. This will become handy for your coming assignment that will come in the next week. So you have to remember those things probably. If I have a promoter, two promoters on a DNA molecule, and I have this is the gene of interest, and I will call this as promoter A and promoter B. Which one produces the antisense strand? If I ask, what will be the answer? Someone can answer it. The antisense, if promoter A is activated, you would get everything in AUG and TGA. UGA. I hope you realize that the orientation of the ORF is given this way: start codon to stop codon. So this would have been the sense. When B is activated, the promoter B is activated, you would actually get the antisense of it. You would read from 5' to 3' here, right? It will be the opposite of UCA and something like that, CAU. Okay. Yes. So what I'm trying to do here is we have a fragment, a gene of interest. This is the gene of interest. It does not have any restriction sites at the edges. There are no restriction restriction sites. And I want to clone this fragment into a vector that has in a directional. This is about directional cloning. Okay, you have to remember that. I think you all know this, right? If I had a restriction site, and that is a vector with a restriction site BAM-H1, and I have a fragment, I will actually draw it in one orientation so that you have it like this, and this is a BAM-H1 site, and here is another BAM-H1 site. If I mix these two and recombine these two, adding ligase after processing with restriction endonucleases, I would get two possibilities or three possibilities. One is the vector religation. The another one is I would have had the ORF ligated like this, and in another case I would have had the I would have had the ORF ligated in this direction. So I'm giving you a situation X, Y, and Z. There are three possibilities I'm giving. There are other things also. The first question is, how can I reduce X? X is absolutely, it's actually a complication to our goals. I don't want X. What should I do? Anybody? We have learned something in the last class. I want to vector religation using alkaline phosphatase. We have to treat the vector with alkaline phosphatase so that vector religation is inhibited. What is the desired? There are now X and Y. There are two possibilities, out of which this is useless because we want to clone, we want to express the gene and see what is going to form. But here, if you have it in this direction, then it is not useful. We want to have it only in this direction, right? So if we want to have it, the problem here is we only have one restriction site. Instead, if we want to have it directional, then we can use, we need to use two restriction sites. Say, for example, here is EcoR1 and here is BAM-H1. And I have here EcoR1 and BAM-H1. Then I can cut these two, assume there is a promoter here, and you can directly clone them. And what you will get is promoter plus the ORF in the proper orientation based on the EcoR1 site and PAMH1 site. That is fair and simple. The problem arises when we don't have any restriction sites here. Oops, sorry. What happened? I think it got erased. Okay, that's all right. So we have the the fragment, and it does not have any of the restriction sites on the edges, but our goal is to clone it in between EcoR1 and PAMH1. Then what we can do is design primers, adding linkers to the primers. Those would have been called as cloning primers. Because, and we are also trying to do it as directional cloning. That is another part. So now I am taking a sequence, ATG. I think I have another sequence here. I will use this sequence, and I will give you a work. We can all work it out together, where our goal is to clone. This is the vector, and here is the promoter, and I have two sites. One is PVU, PVU1, and the other one is KPN1. Okay. Now I have given here one sequence. I hope you can see that this is the start codon. Then you have second, third, fourth, fifth, and then you have the stop codon. Right? It will be good if you can also write it along with me on a paper if you have. So I'm writing the complementary strand. I will write it a little in the bottom. So I have C, G, A, T, A, C. So this is the 5' and here is the 3'. I need to make primers, and the objective is to clone this fragment this much into and in the same orientation of where it would be start and stop codon here. So for that, what we should do 3'. Any polymerase will only add here ATG. I am writing as if you are stuck with primer designing, we will do it again later. Let me finish this cloning first. And if I used, so this is 5' and ATG, that's a 3' base primer I have chosen, so that polymerization would have occurred in this direction. I should also choose one for one on this side. So I will choose something here. So TCA, and this is the 5' of it. So this is the 3' base, 3 base primer. If I added these two primers and added DNA polymerase or Taq polymerase and everything else, what I would end up will be multiple sequences of ATG and several sequence and TGA. And its complementary strand. something like this. Now I cannot use this to clone here because it's not compatible with PVU1 and KPN1 overhangs. So what I should do is I should have added something else here so that it can be cloned here. I can do that in simply. First of all, I want you to remember that primers are not this small. They are usually about 20 to 30 as the need may based on the need, but they are not those so small. For concept's sake, we can assume that they are small like this. Now, what we can do is, what if I wanted to have a sequence PVU? The sequence is CGATCG. I will add a sequence here. CGATCG. This will also, so if I have to add, what I will need is, if I want to add this fragment into this vector in this direction, then I must have had a linker, something like this. If I add linker now, typically the problem is if it is a tucked DNA polymerase, you will have an additional A, like we have seen in the DNA in the TA cloning. So it is not easy to add something directly like this. And if I add a linker like this after PCR, you would get the linker on both these sides. That's not what I want. I want only on one side. I need to have PVU. On one side, I want PVU. On the other side, I should have KPN1. And the sequence of KPN1 is GGTACC. Right? So this is KPN1. How can I get a amplified product such as this one? And there is one more thing you have to remember. If I gave to EcoR1 or BAMH1, if I gave a nucleotide like this, a oligonucleotide, just this is the recognition sequence, and I added BAMH1 here. It may not be able to cleave efficiently. It's because there is not enough space. BAMH1 or most restriction enzymes would also require that there is some more new bases here. It doesn't have to be a particular sequence, but something must be there for the restriction enzyme to interact with this and make the cuts. Okay, so if you look at this is not required for all types of restriction endonucleases, but most restriction endonucleases require somewhere between 3 to 6 base pairs addition on both these sides. Okay, just remember that. So now I want to, if we add PVU to this sequence here, it might not be able to cleave it efficiently because on one side it has the DNA here, but on the other side it does not have enough buffer sequence. That's what our lab used to call. You can call it anything. I hope you understood why we need to have some additional sequence on both these sides. So if you want PVU1 or KPN1 to cleave it very efficiently, we should have had some more sequence to it, something like this. You can have, I'm just making up some additional sequence just like that. Hello, can you hear me?
Yes, sir.
Okay, where was I? So, yeah, I think I'm here. And then I had the sequence specificity. I mean, in the primer, this is for sequence specificity, the target sequence specificity. I want to amplify this region, so I have used a bit of that. I also wanted to have a PVU site, PVU1 site, so I added in this.
Sir, sir, the screen is not shared.
Oh, sorry. Okay. So
Yes, sir.
yeah, this is the sequence that we are looking at to amplify. That's why we added this region here. And then we have added the second part of the sequence in the primer, that is the restriction site. And then we are also going to add some buffer sequence or whatever. Why do we add this? This is for stable interaction of the restriction enzyme to the sequence, because the restriction enzyme needs on both sides some sequence to hold on to it properly. Let's try what we would do with the on this side. Here again, I want to add KPN1. Writing for this would become tricky. This is 5' end and this is 3' end. And now you have to write something for KPN1. If I wrote for KPN1, if I wrote as a KPN1 sequence is GGT ACC. If I wrote the sequence here, GGT ACC, is that right or wrong? It would have been, here would have been 5' and where is it? And this would have been the 3'. And that is not how one should read the sequence. Sequence should have been 5' as in KPN1 here, 5' to 3', GGT ACC. So this sequence is actually wrong for that restriction site. So we need to actually put the complementary strand of it, something like, I can write it this way instead, GGT ACC. That's KPN1 sequence. And I can add some buffer sequence like this. Okay, you don't have to call it buffer sequence. That's how I learned it, so I'm just using it. So here we have 3 parts again to this primer. One part is the sequence specificity, homology to the sequence that we want to amplify. The second part of the primer is the restriction site that we want to add to the onto this end. And the third part is the buffer sequence to increase or to allow stable interaction of the restriction enzyme with the fragment that should be cut. So what I'll do right now is I will try to erase this and make what will happen if we do PCR of this. Okay. So assume that now PCR is run. What will happen in the first round? So from here, I will write the sequence. CGG I wrote, that is for buffer sequence. Then I have a sequence CTG, CG. Then I have ATG, that is the sequence of gene of interest. Then I have a stretch of nucleotides. And then I'm going to have TGA. And I will continue a little bit like that. I'm writing this sequence, okay? I'm trying to write only the first round of PCR reaction. And the remaining rounds you will have to do, but this is how I'll go. The complementary strand of it, if assume it happened in the same place, it would have been, this is ACT, and then you would have CC, this one, CCA, TGG, and then I have GGCG. So this is the buffer sequence, and here you have the KPN1 site, and this is the sequence. Here is the buffer sequence, this is the restriction site for PVU1, and here you have the sequence of it. Then you will have this DNA, and then you will have the region like this. If another round of PCR has happened, you will over the period of time, what you will get is a sequence that is ATG, then TGA, ACT, CGCC. Sorry, this is G, right? Yeah. And then you will have a sequence like this. This is what your ultimate, eventually the product will look like. This would be the sequence that we are actually trying to amplify. The rest of the sequences are for constructing a recombinant DNA. The idea was to construct it. Promoter is here. Here you have PVU1 site, and the other one is KPN1 site. And you have the second part of it. That part is the restriction. Now you have, because you added it in the linker sequence in the primers, now you have a product like this. One side you will have KPN1 site. The other side you have PVU1 site. CCA, TCG, is that right? CGA, TCG, that's right. And on the other side, you have GGT, ACC. And we also have a small sequence of nucleotides because we want efficient interaction of the enzyme to these sites. That's why we have another, I'll call it a buffer sequence or additional sequence, you can say. Okay. So when we, the idea of this is for you to be able to design primers. Why I'm going into this slow is that I will try to discuss again this one in the next class, but eventually not in this week, but the coming week, you will have an assignment. You already have a sequence that you worked on for the molecular biology revision. There you will have to design primers. I will give you a different I will tell you what restriction endonucleases should be for each of you and what should be cloned. And then I will also give you the vector, and you will have to design 2 or 3 ways of cloning it. Sometimes I might say with PCR you have to do, sometime in one strategy without PCR you have to do. So there will be 2 or 3 strategies when you finish an assignment. It's again video-based assignment. When you do that, I think you would have understood a lot of these things. So I will just stop in 2 seconds. Now this fragment is generated. I would have treated the vector with PVU and KPN1, and there should also be, please remind me about this, buffer compatibility. And I hope you know double digestion. Okay, remind me if I didn't connect this. I will just describe this so that it will be useful to you. Anyone who is ever going to pursue research in biology will have to clone genes at one or the other point. If you have to clone and you having good understanding of, or the theory of the standard cloning protocols and logics, if you know that, then you will be an added advantage to the lab, because they don't have to teach you everything. All they have to show you is, okay, here is the enzyme, just add and so on. So in this course, you will try to become good in understanding the theory of it. We don't necessarily have to do the lab in the lab ways, but we will see things much more clearer on the paper. So when you add PVU1, this one would have cut, sorry, here. It will cut here and also here, like this. And when we add KPN1, it would have cut here and something like this. So here is a 3' overhang. Is that right? Yes. 5', 3'. This is 3 and this is 5'. And here KPN1 would also have generated a 3' overhang. Okay, so once we are able to do that, we will get this fragment. We will perform electrophoresis and remove all the remaining parts, like these parts. All these parts should be excluded, because if they are there in the reaction, they will religate to this part, to the fragment. If those kinds of things happen, they will cause they will reduce the probability of recombinant DNA formation. So we need to purify the fragment after digestion, and then we will mix it along with the processed or digested vector, and then we will have the recombinant DNA in the proper orientation. The start codon is on the PVU side, and the stop codon is on the KPN1 side. Okay, that is how we would have done it. What I will do is, we lost some time. You can post me your questions on this particular topic. I'll try to make a video for that. That way it will be beneficial for most of you. Okay, the topic is, we discussed about TA cloning, not much of a detail problem there. The problem would be with the most important part today is the restriction or the directional cloning using primers, how we can using PCR, and the important part of it is the primer designing. Okay, any questions? Or please post me your any questions and make a list so that I can discuss those in the next class. Okay, any questions, please? Okay, then we'll see you. I want you. You can WhatsApp the message. You can WhatsApp me the questions as well if there is an issue, and then I'll try to discuss those in details. Okay. Thank you, everyone.